pass this around. You just put your name and the class and the instructor's name. And then if for some reason you don't get a chance to do that, we'll have this up on the table at the end. So thank you.
For those of you just coming in, we have a sign-up sheet going around that you can find, and you can, if you're getting some sort of class credit, you can put your name on it and then the class instructor for that too. Give a sound check. I'm making sure that he has sound coming out. Thanks for coming. I'd like to introduce our second seminar speaker of the semester, Dr. Steve Cresawn. So he graduated from James Madison University for undergrad and then attended University of Florida for his PhD. And went then on to a postdoc in Graham Hatfull's lab at the University of Pittsburgh. And some of you that have taken the phages option, the phage isolation course that can be taken as an option along with Bio 201, might recognize the name Hatfull or Cresawn from some papers you've read when you isolate your phages or look at the genomes of those phages if you took the follow-up course to that, the bioinformatics course. So Dr. Cresawn did his postdoc in that lab with Graham Hatfull and then kind of started this collaboration that's been going on, my understanding is really ever since then, and that has really led into his current work where he is faculty at James Madison University, back to where he did his undergraduate degree. And his lab there is funded by, in part, the Howard Hughes Medical Institute, which is a really prestigious organization that funds a number of things, but a big focus of their work is biomedical research and biomedical education and programs. And so he will tell us about that in his seminar right now. So I'll let him do that, but help me welcome Dr. Cresawn.
Thanks very much for the very kind introduction. I had a lovely day on your campus today, kind of hanging out with some students, got to go to class, got to have lunch with some students and get a tour, and it's been a really good day. My trip to get here has been an interesting one. I come to you from James Madison University, which is in the Shenandoah Valley out in Virginia. So it's a bit of a ways to get here, and so I kind of combined my travels with a trip out to see my brother in the desert in Tucson, Arizona, and we went out to this place called Jerome in Arizona, which is like an old abandoned gold mine. We went to this event for people that have Volkswagen camper vans, and we just hung out in the desert for like 3 days. It was awesome. And a little bit of scenes from my travels. Last night I walked around and I went to the Capitol building and I found this beautiful Volkswagen van parked right out in front of it, and it was like this omen or just something that told me I'd come to the right place. This is actually only my second trip to Idaho. Both are car related. We'll come back to the other one at the end. As you've heard, my work is done in conjunction with a program called phages, which is through the Howard Hughes Medical Institute. So I've had the really good fortune to be part of this program for quite a number of years, starting in 2005 when I began my postdoc working with Graham Hatfull. phages is this giant collaborative science education program. It started with just 12 universities, but it's grown. We're up to 159 institutions now, different universities and community colleges. About 5,000 students a year are taking this class, including some of the folks in this room tonight, which I think is fantastic. There's a network of something like 300 professors that are all working collaboratively on a single scientific project. And the program, which started in the US, is now spreading to other places too. You can take these courses in Mexico, New Zealand, and now in Nigeria as well, and it just continues to grow. So I thought for today, maybe we could just talk a little bit about, what is all this about? Why do you want 5,000 people working collaboratively on a single scientific project? Does that make any sense? And so let me give you my sales pitch for why I think this is a good idea. So what we work on are bacteriophages. Bacteriophages are viruses that infect bacterial cells. So just like you or me, we can get sick with a virus, but bacteria can too. When a bacterial cell gets infected with a virus, the virus makes more copies of itself, the cell explodes, the viruses go everywhere, and they infect more bacterial cells. This turns out to be a really good way to make more of yourself if you are a virus, and the bacteriophages do it better than anyone else. So our current estimates are that there are about 10 to the 31st power bacteriophages on planet Earth. That's a lot. So we think that there are something, according to NASA, something like 10 to the 21st power stars in the universe. So if that's right, then that means there are enough bacteriophages on Earth that if you could collect them all and travel throughout the entire universe, you'd have enough bacteriophages that you could put 10 billion of them on every star in the universe. There are more bacteriophages than all other biological creatures on Earth added together, including all the other types of microbes. They're nature's single most successful experiment. So if you want to learn about things like genomes or DNA, how things work, how traits get moved around in populations, and what makes a successful strategy, the bacteriophages are a really good place to do that sort of thing. This is what they look like. So maybe I'll point with this. So this is an electron micrograph. And these are just schematic drawings kind of showing the same thing. And this is one example of the structure of a bacteriophage particle. There are other types too, but this one is a fairly typical one. The particle has a head, which is this thing. You can see that it's not this sort of flat two-dimensional shape. It's a three-dimensional structure, closely resembling an icosahedron. If you remember your high school geometry, that's the head. This is the tail. The tail is a hollow tube-like structure. Sometimes it's rigid, sometimes it's flexible. It depends on the bacteriophage. And the particle was made entirely of protein, except for the genome, which lives up here in the head. The genome is sometimes made out of DNA, it's sometimes made out of RNA. It can be single-stranded, it can be double-stranded. If you can think of a strategy by which to encode your genome, then somewhere there's a bacteriophage that thought of it first and is already doing it that way. And these things down at the bottom are called tail fibers. These are structures made out of protein, and they're important for recognizing molecules on the surface of cells that the bacteriophage can infect. So they attach to the cell. The virus sends the DNA from the head through the hollow tail into the cell. It's like a syringe, traveling until it finds a cell that it can infect. It attaches, it injects the DNA, it takes over the machinery of the cell and allows it to make more virus particles. and then they go out and do the same thing all over again. So just a quick history lesson about bacteriophages. We just celebrated our phage centennial. So if you didn't know to be excited about that, you have 99 years to get ready for the next one. They were discovered in 1917 by this French-Canadian guy named Félix Durrells, who was not some big fancy scientist working at some top-tier university. He was, in fact, just a regular guy who was kind of interested in how stuff works. He went to high school, but he had no formal education beyond that. Despite that, he was nominated 10 different times to win the Nobel Prize. He was 0 for 10. He should have won, but he didn't, mostly because he wasn't well liked by his peers. He was kind of, well, a nice way of saying it would be that he was difficult to get along with. He didn't make a lot of friends. He had a lot of arguments with other scientists. He got into it with the department chair, and that didn't work out well for him. But it was sort of interesting. He went from finishing high school to landing a job at the Pasteur Institute in Paris, which is quite the place to be for a budding microbiologist. And it was on the basis of discoveries like this one. So I know that's kind of small, and I could have made this shorter, but there wasn't any part of this passage that I wanted to leave out. So I'll just read this to you. On opening the incubator, I experienced one of those rare moments of intense emotion which reward the research worker for all his pains. At a first glance, I saw that the broth culture, which the night before had been very turbid, was perfectly clear. All the bacteria had vanished. As for my agar spread, it was devoid of all growth, and what caused my emotion was that in a flash I understood. What caused my spots was in fact an invisible microbe, a filterable virus, but a virus parasitic on bacteria. Another thought came to me also. If this is true, the same thing will have probably occurred in the sick man. In his intestine, as in my test tube, the dysentery bacilli will have dissolved away under the action of their parasite. He should now be cured. So Durrell's own words, describing an experiment that he had done, which is like ones that you might have done in a microbiology class. He was growing bacteria in a flask. He added his bacteriophage, and the flask went from being really turbid or cloudy to being clear. And what that meant was that the cells in the flask had died. All the bacteria had been killed. And he understood that what was killing them was not some chemical agent, it was a virus because it could be filtered and then propagated from one round to the next. So it was making more of itself. That was the key insight. He also, remember, worked at a time when maybe the regulations on doing clinical trials in humans were different than they are now. And this was right in the middle of World War I. There were a lot of people getting sick on the battlefield. And Durrell became involved, and he decided he would do experiments on soldiers who had developed dysentery. So unsanitary conditions in barracks, and a lot of people crowded in together, and he worked out ways to culture the bacteriophages from the dysentery bacilli that was in the diarrhea of people who were dying from these types of infections on the battlefield. So he did a kind of not carefully controlled clinical trial, but clearly one that, at least to his mind, was successful. While he was at the Pasteur Institute, he became good friends with this guy named George Eliava. And Eliava was from Georgia, not like Atlanta, but the other Georgia, the one that was in the Soviet Union. And Eliava wanted to go back home. He was a famous microbiologist in his own right. He had founded something in the capital city of Tbilisi called the Tbilisi Black Death Center, which I think would be a great name for a metal band. but it was a research center to fund work on the bubonic plague. And Abba became fascinated with bacteriophages from his conversations with Durrell. So he recruited Durrell to go back with him. That's a little hard to see, like impossible to see. Just to orient you though, here's Italy over here. This is Turkey, and Russia's up here. Georgia's kind of right here in between the Black and Caspian Sea. Tbilisi's the little red map marker. And they went back kind of late 1930s to set up a bacteriophage research institute. So World War II was kind of upon us. There's Adolf Hitler and some other Nazis. Here's Joseph Stalin. Himself not a great human, you know, in his own right. The enemy of Hitler does not necessarily mean he's your friend. Stalin had a buddy named Beria, who is the head of his, what we would call the Secret Service. Beria's chief job was to protect Stalin from being murdered. He was the least savory person under Stalin's command by many accounts. He was a serial rapist and murderer, and he was part of Stalin's purge of intellectuals during his time in power. So there was this thinking that intellectually advanced people were a threat to the society, and they wanted them gone. There's also a rumor that Beria and Abba were both in love with the same woman, and it's not really clear what the reason was, but Beria had Abba killed. Durrell fled from Tbilisi, and the center that they had started was sort of left to wither and die, and Durrell moved back to Paris and never returned to Georgia. But nonetheless, the center didn't die. They switched gears from being more of a research-oriented place to one that produced bacteriophages on a custom basis for sick people to treat bacterial infections. So when we think of bacterial infections, we think antibiotics. That's the way that you treat them. But that, for many, many years, that was not the primary mode of treatment in Georgia, in Ukraine, and in some of the other Soviet states. The bacteriophage was. So here on the left, you can see some vials. I don't know if you can read the writing. It might be you can't read it because it's too small, or it might be that you can't read it because it's in Russian. So bacteriophage therapy was a huge deal there, and to this day, it still is. And you'll occasionally hear news reports of famous, wealthy Western celebrities with these difficult-to-treat bacterial infections flying to Tbilisi and getting a custom phage therapy treatment. They've been doing it successfully for 100 years. The vial on the right says investigational product for clinical trial use only. That's one from the United States, where we are just now getting around to take this idea seriously. So the problem here is antibiotics, right? Antibiotics were discovered in the 1920s. Alexander Fleming gets most of the credit for penicillin, deservedly or not. He comes up with this idea of penicillin, which rapidly gets commercialized and mass-produced and used to treat people with bacterial infections. On this slide, you can see, if we just ignore the anti-malarial compounds at the top, you can see antibiotics. When they were introduced on the left, when resistance to those antibiotics was first observed, and when they were essentially abandoned for newer forms of antibiotics. So in most cases, antibiotics only work well for a few years clinically before resistance arises. And there are a lot of different reasons for that, overprescriptions being one of them, people not necessarily understanding the differences between bacterial and viral infections. But another big part of the problem is agricultural use of antibiotics, where they're used as growth-promoting factors in livestock and in poultry. And so they're everywhere. Antibiotics are all around us, and the resistance problem is encoded in the DNA of the bacteria. Bacteria do what they do. They spread genetic material from one cell to the next, and antibiotic resistance moves through a population until it makes it into humans, and we are left with a situation where people have infections that can no longer be treated with at least the typically used antibiotics, and in some cases can no longer be treated with any antibiotics. This is especially a problem with tuberculosis, where multi-drug resistant or extensively drug-resistant TB, there's really no therapy left for those patients. In some parts of the world, XDR, TB, and HIV/AIDS substantially overlap in terms of the affected patient populations, and studies there have shown that from diagnosis with TB to death can occur in as little as 2 weeks, where in the West, many people live with TB with no symptoms. And it's thought globally to affect about a third of the world's population, but very few people as a percentage of that succumb to it. Here's another problem. Pharmaceutical companies, they're not really looking for new antibiotics like they used to. And the reasons for that are mostly financial. Sell someone an antibiotic and they'll take it for 7 days, 10 days. But if you sell them an or a high cholesterol drug, or a drug for diabetes, or a drug for high blood pressure, they'll take it every day for the rest of their life. There's a lot more money in those types of drugs than in antibiotics. And so major pharmaceutical companies are essentially giving up on the search for new types of antibiotics. So the old ones are not working anymore, and no one's looking for new ones. That's a bad combination. The World Health Organization is warning that we're headed toward a post-antibiotic era, an end to modern medicine as we know it, where things that used to be routinely treated will become fatal. I think this is maybe slightly too fatalistic for where we are in time right now, but if we don't do something, the direction that we're headed in means real trouble. So I want to tell you a story. I was going to show you a video, but it doesn't have any sound, so I'm not going to show you the video, but I'll just tell you the story. In 2015, this guy there on the right, Tom Patterson, and that's his daughter on the left. We'll come back to her in a second. But this guy, Tom Patterson, and his wife, Stephanie Strathy, went on vacation to Egypt. They went to see the pyramids, do all that stuff. They went out to eat. Tom got really sick the next day. He had a really high fever. He was vomiting just uncontrollably. Stephanie thought he had food poisoning, but she had a buddy that was an MD back at home in San Diego, and she texted him and was like, "Hey, this doesn't seem right. I'm kind of worried. I don't know what to do." She actually had a lot of buddies who were involved in medicine because she was the dean of epidemiology at the University of California in San Diego. And Tom also had a bunch of the same buddies because he was a faculty member there as well, studying HIV and AIDS. He's actually a psychologist studying the psychological impact of the AIDS epidemic. And so she was pretty well connected, they both were, and they arranged to have him medevaced from Egypt to Germany and then back to San Diego, where they discovered that he had an abscess in his pancreas that was filled with a bacterium called Acinetobacter baumannii, which is a really substantial public health problem in the Middle East. And it's one of the main things that affects soldiers who are U.S. soldiers that are returning from that part of the world. And they'd given him all the normal antibiotics and nothing was working. He slipped into a coma. He was in a coma for a couple months. They thought he would die. They were sure he would die. But Stephanie came up with this idea that they should explore this therapy that no one really knew much about at the hospital, which is called bacteriophage therapy. And the physician who was treating him said, "Sure, why not? There's nothing else we can do." And so she started just going on Google and looking up people that study bacteriophages and emailing them. And she got in touch with a guy named Ra Young, who's a professor at Texas A&M University. She got in touch with people at a company called Advanced Phage Therapeutics and some folks at the United States Navy who study bacteriophages as a therapeutic for sailors returning from other parts of the world. And all these people started saying, well, we have some phages in the freezer that infect that particular host. And so people started sending phages from all over the world. They mixed them together, they put them in an IV, and 2 days later, Tom woke up from his coma. He'd been in the coma for 2 months, He woke up, he recognized his daughter, and within a matter of days, he had recuperated enough that he was able to go home from the hospital. He's now essentially fully recovered, having lost about 100 pounds in the process. He says don't do this as a weight loss program. But it was a very high profile case for phage therapy working successfully. And it started this sort of mission to connect phage researchers with physicians to be able to treat patients in a super timely way with a bacteriophage. So this is a project called the Phage Directory, which essentially is just that. It essentially connects people over Twitter. So if you're a phage researcher, you can go on the site and put in information about what phages you have or what hosts they infect, and then you'll just get a tweet that says urgently needed, bacteriophages that infect Acinetobacter baumannii or whatever it might happen to be that day. And because of the success of this project, Stephanie and Tom and Chip Schooley, who's the physician at UCSD, have set up the first bacteriophage therapy center in the United States, in San Diego. So this is sort of a growing trend to do this type of work of connecting sick people and their physicians with bacteriophage researchers. And the tricky part is that unlike antibiotics, which are like the nuclear option for treating a bacterial infection, bacteriophages are highly specific, right? You all know from taking antibiotics that if you read the label, it says like may cause stomach upset or may cause all other kinds of infections. And it's because antibiotics aren't very specific. They take out lots of bacteria, including the ones that live on us and in us and keep us healthy, our commensal bacteria. But bacteriophages are highly specific. So they do a lot less collateral damage, but the tricky part is it's pretty hard to find the bacteriophage that you need for the particular strain of the particular bacterium that you might be infected with. Hence the need to sort of crowdsource this type of thing. What you need are thousands of phages in a freezer somewhere. That's why the C-phages project is so impactful. We have about 10,000 of these viruses sitting in a freezer, and when a patient says they have a particular infection, there's a reasonable chance that there's a phage in the freezer that can be used to treat it. The interesting thing about these phages, though, is they come in all different shapes and sizes. They've got all kinds of different genomic features. And unlike a little molecule of penicillin, the bacteriophage is complicated. It's made out of tons of different proteins. It's got about 100,000 base pairs of genomic information and somewhere between, say, 50 and 350 genes in its genome. If you're responsible for regulating new drugs, you work at the FDA, and someone comes to you and says, we'd like this to be a drug. It's got 150 genes in it. We only know what 20 of them do. But we think it kills the bacterium that is making this person sick. You probably have some concerns, right? Well, what if there's other stuff in there that's bad? Well, that's why it's important that you sequence the genome and that you have tools to study the genome to try and learn about what all those genes do. So most of the information, most of what you need to know about the bacteriophage, you can't get it from a picture. Although these are great and they're helpful, these kind of transmission electron micrographs. But the good stuff is what's hiding in the genome. Fortunately, genomics is getting much cheaper to do, right? So this is sort of just a quick look over the last 15 or so years at the cost of sequencing the human genome, which goes, from hundreds of millions of dollars to do the first one, down to, they're saying now, like something like $1,000 to sequence one genome, and the price continues to fall. It falls much faster than Moore's Law, right? So Moore's Law is this idea that about every 18 months, the technical ability or the speed of your computer doubles. So we think of technology as like this sort of inexorable advance, and it's just faster and faster and faster all the time. sequencing is getting faster at a much higher rate than the phone in your pocket or your MacBook. So what happens when you sequence tons of stuff is that you get a lot of data. And most of the people doing the sequencing, like everyone's doing sequencing now, it's really cheap and it's easy. It produces tons of data and it creates problems that maybe aren't the most familiar for biologists, right? You're used to using pipettes and you're used to maybe cloning some genes and running some gels or whatever. You're not used to like architecting a database or figuring out how to set up cloud computing or these types of things. But these are really becoming fundamental schools, not just for fundamental skills, not just for biologists, but really for anyone that wants to do science, because a lot of the sciences are headed in this direction of big data. And so data science is this, fairly new term. It's been said like data science is the new oil, if you want to believe that. The one that I like better is data science is the new soil. It's this sort of fundamental thing for growing new ideas. And the idea is that it's an interdisciplinary set of skills involving how you approach problems, things like algorithms, but also how do you visualize data and derive meaning from tons of complicated information. So what I work on are, in large part, are data visualizations. This is my favorite data visualization ever. Just give that a second to sink in. What's that?
Have you got to eat the pie?
No, unfortunately, it's not my picture. I really wish it were. But when you think about data visualizations, maybe you're used to thinking about things like pie charts and scatter plots and stuff that you can make in Excel or any other spreadsheet program. And those are great. There's nothing wrong with those tools or those types of visuals. But a lot of times you want to create custom visualizations of some data set for which there is no like insert chart button that does this in Excel. Like there isn't insert comparative genome map in at least the version of Excel that I have. And so what my lab has been working on is trying to think about how can you create your own visuals to help discover the cool things in your data set and be able to tell the stories of them. So this one's not mine, but it's still awesome, so I thought we should look at it. So this is at the Wall Street Journal, Battling Infectious Diseases in the 20th Century, the Impact of Vaccines. So it probably doesn't all quite fit on the screen, but this is measles. So every row here is a state, and every column is a year. Unfortunately, not all of the states get a label because they didn't make the picture tall enough, but they're on there. You'll notice that Idaho did not get a label, but Virginia didn't either. So I feel your pain. But you can hover your mouse over these things and see for every state and every year how many cases you get. And they've also helpfully drawn this line to indicate when the vaccine was introduced. So all these little colored dots are a heat map that just simply show the number of cases in each state and each year. The point of the visual is to illustrate the effect that the measles vaccine had on the number of cases, and not just in one place, but essentially everywhere. Here's hepatitis A, for which the data does not go back nearly as far, and the vaccine is much newer. The trend is the same. Here's the mumps. There's no pre-vaccine data, unfortunately. Pertussis, where they took a decade off from collecting any data. Here's polio. Same kind of deal, right? Number of cases is going up, up, up. Vaccines are introduced, and the number of cases goes to something approaching 0. In fact, in the US, it is 0. And we don't need to look at all these. They essentially tell the same story. All right, so we made a kind of similar visual to this to help us to analyze phage genome data. And the thing that we built is called famwriter. The name is kind of dumb, and I won't bother to explain where it came from. But it's a computer program that draws maps of genomes, interactive maps that let you explore phage genome data. And it looks something like this. But it looks really weird because of the size of the screen. But anyway, the idea here is that we've taken all the phages, we've run them through some analyses, and we've put them into these groups that we call clusters. Every one of these groups is a cluster. If you click on it, These are all the phages that belong to that cluster. You can see there are quite a few. The ones that start with A are the ones we found first. We found them first probably because they're the most populous in nature. So these A ones tend to be big. If you scroll down towards the bottom, you find ones that have fewer in them. That's my brother's name. So what can you get from doing something like this? Well, we wanted to be able to draw pictures to help us to analyze the data. And the pictures, they look something like this. Let's zoom this out a little bit. So these two phages on the top are called BPS, maybe I'll point with this, BPS and Halo. Bacteriophages are cool. You can name them whatever you want when you find them. So how many people do you know that have found their own organism and gotten to name it? Well, about 2,500 that found bacteriophages. And what we're looking at is a comparative genome map of these two phages. These little colored rectangles show genes in a genome. So these are the genes in BPS. These are the genes in another phage called Halo. These are the genes in an unrelated phage called LeBron. There's a story there too, but I don't have time to tell it. The color-coded stuff between the genomes, that shows how similar they are at the DNA sequence level. And you can scroll through and find cool stuff in the genomes, like areas where there are sequences that repeat and show up with these kind of crazy-looking X patterns. Here's another one where a region's in the genome 3 different times. You can put your mouse over stuff, and it'll give you the statistics that show how related they are, which is really useful. You can get all this same data by looking at a table, but it would be 10 feet tall, and it's very hard to extract the meaning from it without a picture that represents it. In fact, we worked with these phages for years before we had this picture, and we never noticed this region right here, which is a gene in one place in one virus, this one, and if you follow the line, it's in a different place in the other virus. That turns out to be a transposon. It's a mobile genetic element, something that can move on its own accord in the genome. And it's really tiny compared to other ones that were already known. In fact, it's the smallest yet described mobile element in any kind of organism. And we had the data for years, but until we had the picture, we didn't see that it was there. So what my lab does then, essentially, is we just draw pictures. We make software that draws pictures, and it looks sort of like this. And what I thought that we could do for maybe the last 20 minutes or so is to talk about how you do stuff like this. So this is, I would call this a sort of fancy version. You can click on things and get DNA and protein sequences and information about what the genes do. You can do things like click and drag the genomes and reorder and recompare them. You can move them to align things however you think they should be. And all that stuff takes years to get right. But what I wanted to do was just start at the beginning. And for those of you that maybe you've never had any programming, you never had a class in computer science, learning how to code seems like just as hard as learning, I don't know, a foreign language. And so maybe you just didn't want to go there. But my thinking for now is that for most of you, you'll sit in the room for the next 20 minutes because someone told you it would be rude to leave the seminar in the middle. And so for that reason, I can have you like my captive audience and show you some programming stuff. And maybe by the end of it, you'll say, oh, that's actually not really that scary. And maybe a little bit useful, or maybe a lot useful. Okay, so that's my plan. We'll see how it goes. All right, so we're going to talk about two things. One is called the developer tools that are built into your web browser. And the other one is a software library called D3. So, oh yeah, we were shopping for a Volkswagen van. I think I'm hooked. So if you go to the little menu button in Chrome or whatever browser you like, more tools, developer tools, you'll find this like secret thing that's been hiding there all along that maybe you didn't know about, and in it is a console or a place where you can type commands and see the result. And so we're going to type some commands in a language called JavaScript. I don't know if you've heard of JavaScript before, but it's the world's most popular programming language by most estimates. And it's the fundamental language of the internet. So any website that you visit that's in any way interactive, it's running JavaScript code behind the scenes. You don't see the code, you just see the output of the code, which are the things that are on the web page. Does that make sense? All right, cool. So like any programming language, it does basic stuff that you would expect. You can do math, right? You can do any, simple math. You can do addition and subtraction and multiplication and division. But you can do more advanced types of math too. We're not going to, but you can. It has things that might seem familiar to you from math, like variables. So, x equals 3 means let's make a variable. We'll call it x, and we'll assign a value to it of 3. Cool. And now I can say like x times 4, and it's 12. So it's put the value into the formula that I typed. All right, that's enough math. There are other types of data. There's a type of data called a string. I'm just going to make a string. I'm going to assign it to a variable called a. We'll just pretend it's your address. Okay, I'm telling it it's a string because I put it in quotation marks. That's it. A string is any collection of numbers and letters you want it to be. Usually in code that I work with, strings represent things like DNA sequences. Okay, but they don't have to. It can be whatever. All right, we've done 2. We're going to do 2 more. So we're halfway there. These are simple ones, numbers and strings. You can also have more complicated types of data. One is called an array, which is simply like a thing that holds more than one thing. So I'm going to make an array. I'm going to call it my array. And I'm going to put some stuff in it like this. Okay. So there's a variable. It's called my array. It's got a value. In this case, it's a range of 5 things. Some of them are numbers. One of them happens to be a string. It doesn't matter. You can put whatever you want to in there. You could put an array in there. Ooh. Yeah. It's just a holder for stuff. An arbitrary number of things. And then one more thing, and then we're going to be done with talking about types of data. And this one is called an object. So if we were to think about like a gene as an example of an object, maybe it would have a name. We'll call it like gene 1. It's going to have a start coordinate. Like where in our genome does this gene begin? We'll say it begins at base pair number 30. It's going to have a stop coordinate, say base pair 100. And it's going to have a direction or orientation. Like is it a forward gene or a reverse gene? So DNA, right? It's a double-stranded molecule. It's anti-parallel. It looks like this. It's a weird gesture. This is like I Dream of Jeannie, if any of you remember that show. Okay. So the forward genes are going to the right. They're encoded on the top strand of DNA. And the reverse genes are encoded to the left. They're going that way. So we'll just say forward or reverse. All right, cool. So we made an object. Notice how the object is just a set of paired data. So like name has a value 1. Start has a value 30. Right? So it's like paired things. Name and a value, start and a value, stop and a value, and so on. And I just made these up. I could have called them whatever I wanted, but I picked names that sort of make sense. So far so good? This would be like, now is the part where like, if you're like, I have no idea what you're talking about, please just say so, because otherwise you'd be lost. Also, sometimes your developer tools just spew all kinds of useless, irrelevant information, so ignore it when it says stuff like that. I always do. Okay. All right, so our next thing then is we're going to take this little bit of knowledge that we have and we're going to put it to use to make our own data visualization. All right, so this is a weird website. It's a website that allows you to make a website. And it's got some code in here. This kind of code is called HTML. It's just a way to describe the structure of a web page. We don't need to get too hung up on this, but it has what are called opening tags and closing tags, which just means like a pair of things. So like head starts this section called head, and slash head ends it. And inside you have like script and slash script and so on. So it's like an opening thing and a closing thing and relevant stuff in between. So we don't really need to worry about this section. Body is where all the good stuff is. So I like this because it's kind of like a phage. Like it has a head and it has a body or a tail. Same kind of deal. And then in here is a bunch of code. This is JavaScript code, but it's not written in the console. It's written as a program. So with what we saw before, I typed one line and then it ran that line and showed me the output. Here I'm going to type a whole bunch of lines and then we're going to see the output. In fact, it's what's up here at the top where it says edit the code below to change. It actually says change me, but it doesn't fit on the screen, but that's okay. So what we can do is we can go down here and write our own code that changes some of this stuff. So here's what they did, right? They said create a variable. I didn't type var, you can, when you make a variable, it doesn't matter. Delete this and it doesn't make any difference. But they made a thing called SVG. SVG is just a kind of picture. So normally when we think about pictures, you maybe think about like a picture that you take on your phone. When you take a picture on your phone, the camera is measuring things in pixels. It's got like 12 megapixel camera, so there's 12 million pixels, and it's saying what color light should I put at this one dot of the 12 million dots that make up the picture. That's cool, except that if you like zoom in on part of the picture, now it doesn't have enough data, and so it looks kind of jagged or pixelated, which is no good. SVG gets around this problem in a way. It's just not designed for a photo. It's designed for things more like a diagram or illustration. So it doesn't measure stuff in pixels. You describe what you want your picture to look like in terms of things like shapes. So I wanted to make a rectangle, and it starts at this position and it ends at that one, and it should be blue, and it's this tall or whatever. Or I want a circle, and that's all you say. And then if someone zooms in on it, well, it still looks perfect. It's kind of like how you can zoom in on a PDF file and the fonts all still look really nice. Same concept. So we're going to make an SVG picture, and the cool thing about SVG is it's just like the HTML stuff that we talked about in the beginning. It's just a bunch of tags. So like rectangle / rectangle or circle / circle, and the stuff in between is just like what color should it be and where should it be on the screen. Okay. So what they've done here is they've appended a tag called text to the SVG. They gave it this actual text, what it's supposed to say on the screen. They set an X and a Y coordinate and the font and whatever. We don't want that. We're going to get rid of that. And what we're going to make instead is an array of genes. So it's an array, and it's going to have some objects in it. Right? We know it's an array because it has these square brackets, and the objects, just like before, are going to be these key and value pairs. Right? So obviously this is made up data. I'm just typing it in, but it doesn't matter because you can do the same thing with the real data. And I'm just going to copy and paste it a couple times so that we'll have a few different genes to look at. I'm going to give them each a unique name. Say this one goes from 30 to 100, and this one will go from like 130 to 200, and this one will go from, I don't know, 330 to, oops, sorry, 230 to 300, and 330 to 400. And just for good measure, let's make one of them a reverse gene, so they're not all the same. Cool. So now if we go up and look at our picture, where's our genes? We wanted a map. They're not there. It's because we didn't tell it how to draw the map. We just told it the raw data. So what we're going to do is we're going to tell it, for every gene in this array of genes, I want you to draw a rectangle on the screen. And the rectangle should be at the right place, it should be the right size, and so on, so that it's a visual representation of the real data. So the way that we can do that is we can say svg.selectAll rectangle. So they don't write out rectangle, they write rect as just an abbreviation, same thing. Okay, so we're gonna do this thing called a data join, which basically means I want to connect some rectangles on the screen to my genes that I care about, the things in that genes array. So the way that you do that is by saying select all the rectangles that might be there, and then bind them to my real data, which is this array called genes. Okay, so now we've kind of connected those two things, the rectangles on the screen with the real genes. There aren't any rectangles on the screen yet, I know. There will be. And what we're saying is that anytime there's a gene that doesn't exist as a rectangle on the screen, then it's like it should enter our scene. So we're going to say, okay, it entered, and now we're going to do something. We're going to append a rectangle. And we're going to set some attributes on our rectangle. So it needs like a Y position on the screen, which is just like how far down on the screen it should be. This is the weird part about SVG is it's like regular math, but it's kind of upside down. Like if you think of a normal Cartesian plot, this is fun. The 0, 0 point would be here, right? The bottom left corner. X gets bigger when you go to the right, and Y gets bigger when you go up. In SVG land, it's upside down. So X gets bigger as you go to the right, but Y gets bigger as you go down. So we're setting a Y value of 100, which means put my rectangle 100 pixels down from the top. Cool. So that's great. All our rectangles need to have a height. I don't know, just make them 35 pixels tall. That seems like a really random number. And they need to have an X value. How far over should they be? Well, here's the tricky part. Like the height of the rectangles, they're all going to have the same height. And they're all going to be on the same level on the Y-axis. So we just picked numbers. But for how far over they should be, that should be based on the data, right? The rectangles need to go in the right place in the genome where their gene is. And the thing in our data set that corresponds to where to put them is that property called start. Like where in the genome does it start? That's where the rectangle should start. So it's going to be different for each gene because each of our 4 genes starts at a different place. So for the X value, we're going to run a thing called a function, which is just a block of code that usually has a name, but in this case it really doesn't even have a name. It's just anything in these curly brackets, it will run for each of our genes. And each time it runs it, it's going to make up a variable and call it D. And then inside this part, which is the body of our function, D will refer to a gene. So the first time the function runs, D is this first gene object. So if I said, what is D.name, it would say 1. If I said, what is D.stop, it would say 100. The second time it runs, D refers to the second gene, right? So each time through, D means a different thing. So the X value then is just D.start. And the other thing that we didn't set yet on our gene is the width. And it's also different for each gene. So we need to do a little bit of math, right? The width of the rectangle is the difference between where it stops and where it starts. So we'll say return D.stop minus D.start. Okay, so if we scroll up to the top, we now have 4 rectangles that correspond to our 4 genes. So we have a visualization of our data already. all of our genes are the same size, so it's kind of a little bit hard to see. So let's make this one a little bit smaller. And now you can see that first rectangle has changed automatically to correspond. Let's do one more thing. Let's give it a color. For SVG shapes, the color is unfortunately called fill, and not color, which I think might have been a better choice. Okay, so by using fill, we can give them a color. Great. Well, that color isn't really encoding information. It's just a random color I picked. Might as well have left them all black. But what if we use the color to represent the direction that the genes are being transcribed so that we can look at the picture and tell? So we could say function D, and then we can do one more concept. It's, I promise, the last one, which is called a conditional statement. We can say if d.direction is equal to forward, then we'll return a color that I like called steel blue. Else, meaning d.direction is not equal to forward, we're going to return a different color called dark orange, because those two look nice together. Okay? And now the colors of the rectangles tell us something. Which genes are forward, which genes are reverse. And you can do all kinds of other things to sort of play with the visual encoding of information. Just a really kind of quick, fun one. Oops. Is you can animate the data. Right? So we've been working on this for about 10 minutes, and we already have like an animated data visualization that is color-coded to the real data. One other thing we might want to do that would be fun, and it'll be the last thing, I promise, is that we could do a little bit of animation here that means something, and it's not just for pretty. We can animate the genes, if they're forward genes, to the right. Right? So if d.direction equals forward, then we'll return the negative, well, let's return 0. We could be a little fancy, but we don't have time to be a lot fancy. Else we'll return like 960, which is the width of our picture. Okay, so now they animate according to the direction in which they're transcribed. The genes that are transcribed to the right move to the right in the picture, the ones transcribed from the left move to the left. So the animation actually means something. So this is how we made our famwriter.org site. We just spent time thinking about it and adding things one at a time until we came up with something that we think is actually useful and that has helped us to analyze what is a really now very large set of data. And obviously, you know, there's more to it, and there's like, how do you keep it all running on the server side and the cloud and whatever. But for us, it was just like starting simple, learning a few concepts, and then each day or whenever we had time, adding a little bit more to it until we got up to the point where we had made a thing that we were really proud of and felt like we could use to analyze the data. So, so I just wanted to show you one more picture, and then we'll call it a day, I promise. That's me in like 1998, I think. My dad decided in 1998 that he wanted to buy a 1996 Mazda Miata M Edition. It had to be new. He went to their local dealer in Virginia Beach, where we live, and they were like, "You're nuts, you're not gonna find one. They've been in the showrooms for 3 years. There won't be a new one." And he looked it up in the computer, and there was one new one left in the whole country. It was in Lewiston, Idaho, where apparently no one wants a rear-wheel drive, two-seater convertible. But they should, because it's a really fun car. So, so my dad and I flew to Spokane and got a ride over to Lewiston, and we bought this Miata, and we drove it back across the country. And there's this little note, that's the back of the picture. Steve, thanks for taking the trip with me. I had a great time. Love, Dad. Don't forget the Mighty Potato. If you ever go to Lewiston, there's a restaurant called the Mighty Potato. It was our first stop on the way to Glacier National Park and Mount Rushmore and all these other sites that we saw as we sort of made our way across the country. So, you know, thanks for being involved in the second automotive-related trip that has brought me to Idaho. It's fantastic. And then I can't end without acknowledging the crew in my lab that has done a lot of the work that I presented to you today. So I won't go through who all these people are, but my lab, like I think your department is set up, it's basically all undergraduate students. We're a much bigger institution than you guys are. We have about 24,000 students, but almost none of them are graduate students. We have like 1,100 biology and biotechnology majors. Not enough get involved in research, but the ones that do have a really good time. So I would just like to end by encouraging you to, if you're really interested in biology, you want to be a biologist, or you want to do something in health or medicine or whatever, seek out opportunities to get involved in your department. You won't regret taking that step, even if it seems a little bit scary. And learn how to code. It'll get you a lot of places, like maybe even like 2,500 miles from where you live, to give seminars. And thank you.
So we have some time for questions, so please feel free to ask Dr. Cresawn anything, and then we'll thank you again.
I didn't catch you. You said that there's that lab in Georgia that's been working with these phages for 100 years.
Mm-hmm.
And are you guys collaborating with them at all?
We don't. We probably should, but so our group has not been directly involved in phage therapy up until like 6 months ago. And some of the stuff that we've been doing, it's been very ad hoc and on a case-by-case basis where someone contacts the group. So there's a study going on that Dr. Hatfull is running involving patients infected with a mycobacterial pathogen called Mycobacterium abscessus. In that particular case, or set of cases, it's mostly patients with cystic fibrosis. Cystic fibrosis patients get this accumulation of mucus in the lungs that they can't clear, and that's a great environment for bacteria to grow in. It's a genetic disorder. There aren't really great treatments for it, so commonly they are recipients of lung transplantations. And when that happens, they're taking immunosuppressive drugs because of the transplant, and it's really difficult for them to clear the bacteria. Mycobacterium as a genus is incredibly resistant to almost every antibiotic, and so there are now phage therapy approaches going on to treat individuals for Mycobacterium abscessus, Mycobacterium avium, and other pulmonary mycobacterial pathogens. But it's been done sort of in isolation from the crew in Tbilisi, who, I don't know any of those people personally, but I suspect that you're going to see much more collaboration between US scientists and Georgian scientists going forward. Part of the trouble is they've got 100 years of literature on this subject. It's all in Russian. So that's the first thing. And then the second thing is that a lot of their publications are individual case reports. So their system for publishing clinical trial data is really different than what we require here with things like double-blind studies. They don't necessarily do it that way. It's just like a patient came into the clinic, we used these phages, and this was the outcome.
So, forgive my ignorance about biology. I'm a computer science student.
Oh, cool.
So how hard it is to create a bacterial phage? How the specific ones that treat people, how hard is it to make?
How hard is it to make a bacteriophage?
Yeah.
So we don't have good tools for making them. Basically what we do is we find them in nature. So students that do this, you can ask, I'm sure, some people who are in the room who are either in that class or have taken it previously, but they go out and they collect samples from the environment, usually soil samples. It's hard to get a measure of how many bacteriophages might be in like a teaspoon of soil, but I can tell you that in a teaspoon of seawater, there are roughly 10 to the 7th to 10 to the 8th bacteriophages, right? So like somewhere between 10 million and 100 million of these viruses in every milliliter of seawater. So you probably could make them in the lab, and actually some people have, but why bother? Like they're just right over there. Just go get one of those. So there are two things about them that are interesting. One is they're incredibly abundant. Like I said, there are more of those than everything else put together in terms of biological creatures. I don't call them living things because viruses technically aren't living, whatever. But one thing is they're incredibly abundant, but that might not actually be that interesting if there's 10 to the 31st power of them if they were all the same, right? But it turns out that they're not all the same, and we've sequenced about 2,500 of them, and we haven't seen the same one twice, right? So they're probably not all different, but there are many, 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 many different kinds. Some are a little bit different, some are a lot different. But because a combination of their genetic diversity and their ubiquity tells us that we should just keep sequencing more of them, because the new information, if you drew a graph of the number of sequenced phages on the x-axis and the new types of genes that you discover on the y-axis, if there weren't very many different types, this would be sort of a curve that levels out quickly. But for us, it's just like this. Like the more we sequence, the more we learn. So why stop? Yes?
So like Mohammed, I'm kind of interested in this research on the computer science side of things.
Uh-huh.
And I was wondering what you recommend for students who want to get involved in this kind of data science, bioinformatics research, and like what could make us competitive when applying to research programs. Like if I don't know what your program is like, but if you were looking for applicants, what would you want to see?
Okay, so the students that take our class, they almost never have any computer science skills coming in. And they do a lot of work on computers as part of the class. They're not writing code usually. They're using code like the site that I showed and some other programs to do the analysis. So they're, by the end of it, they've learned something about just like dealing with large data sets. But no programming skills, no skills in terms of constructing your own databases. So the skills that I look for example, like I'm in my research lab where I'm trying to get students that can help to build the tools. The things that I have found most useful are skills in web development and in Python. So we use Python for everything on the back end, and we use JavaScript for everything on the front end. Many moons ago when I was taking computer science classes, I told the students that I had lunch with today, I took a year of a computer science master's program, and I actually dropped out because I didn't like it at all. But I learned enough there to kind of like keep the ball rolling. My undergrad degree was in biology, and most computer science master's programs, they want you to have a computer science bachelor's degree as like your entry point, right? But I found a fairly unique program that happened to be at my home institution at JMU that said, like, bring your STEM skills from biology or chemistry or astronomy or astrophysics or whatever it might happen to be, and we'll teach you to code so that you can make your science better. That's what I sort of bought into. When I got there, it wasn't that, and so I quit. Like everyone that was graduating was getting a job, like, working for Walmart, telling them where to build their next store or something like that. And that wasn't what I was interested in. But yeah, so Python and JavaScript are the things that we use. There's a huge emphasis on learning R as well. It's certainly like a rising star in biology. I'm not a huge fan of R personally. My favorite thing about JavaScript in the world is that you can make interactive data visualizations, and you really can't do that well in R, in my opinion. There's sort of hacky ways to do it, but I don't really like any of them. So yeah, I want to be able to tell a story around my data set to make tools that let me explore it. And so I love the idea of just latching on to the web. It'd be hard to make an argument that web technologies are not successful or growing rapidly. And so that's where we focus. So we've moved away from our old way of doing stuff, which was like we wrote everything in Linux. You know, it was a lot of command line stuff. It was MySQL databases. And we've moved to doing everything in these kind of like internet-friendly databases like MongoDB. It's the one that we use now. And I don't know. Students come into my lab with they're a biology major or they're a biotechnology major. They have no coding experience. And I just teach them kind of like what we just did. And by the time they've left, they're making like substantive contributions to the code. And they're actually, about half my students go on to a graduate program or medical school, like something sort of traditional biology students would do. And the other half are getting jobs at like startup companies doing data analytics. One recently got a job working for a microbiome company. So we didn't really talk about that, but like one of the really hot areas in biology right now is the human microbiome, which just means what are all the organisms that live in your gut? They're increasingly implicated in a number of different kinds of human health issues. And it goes without saying that if there are bacteria that live in your gut, there are bacteriophages that live in your gut. That's a relatively underexplored area. People are really focusing on the bacteria right now, but I think that's like the next wave of microbiome analysis. Yes?
So you mentioned that there's a lot of other groups working on phages in the US. Have
Mhm.
you gotten to work with groups?
Yeah, so well, our network has like 160 universities, so that's been enough partners for me. But there are other places that are not really part of the phages thing, but they're just places that do bacteriophage research, and we do collaborate with some of them. This has been a really fun thing about making a software tool that I didn't anticipate, is that people just write and say like, hey, I want to use this thing. Do you want to collaborate with me? I have a data set and I want to be able to make maps like the ones I saw on your paper. So like I got an email 6 months ago from someone at Harvard that just said like, no one here does this kind of stuff. I was like, really? At Harvard College of Medicine? She said, yeah, like we want to make these maps and no one's interested in this problem. I have this data set. Her data set is twice as big, more than twice as big as the one from the phages project, which is amazing to me because that's one lab that did this thing it's like close to 5,000 sequenced bacteriophage genomes. And it's not the only example, but it's probably my favorite one, at the moment, of just groups that just kind of get in touch from wherever and want to work together on their genomes.
Maybe time for one last question.
Okay, I was just thinking that there's a lot of Russian international students. I've run into quite a few at this school alone, and this is a pretty small school. It seems like it'd be a really good idea to groom them to work towards going back to work at that place in Georgia because they are already bilingual with English and Russian to be able to get into that depth of literature that they already have in Russian.
Yeah, that's a great idea. Unfortunately, I don't know a single international Russian student on my campus. But if I did, I would go home and do that. That sounds like a great plan. Yeah, we have a lot of international students, but for reasons that I don't really understand, they come from a relatively small number of places, and Russia is not one of them.
So we really appreciate you being here. I know it's a long trip for you to make.
Oh, it's been so much fun.
Yeah.
Thank you guys so much.
Yeah, thank you. Thanks you guys for coming.